So my name is Chelsea Chan, an undergrad in Jennifer Doudna Lab, Department of Molecular Cell Biology and Ecology Chemistry, and the title today is Dissecting How Dicer Dices. There we go. Okay. So in a given biological system, the process of how genetic information is transferred is observed through the central dogma of biology. DNA, the source of the genetic information, is copied to RNA through transcription. Then RNA is synthesized to protein via translation, using RNA as a template. What I specifically work on is RNA, which is the copy of DNA. So like most scientific discoveries, there is some history behind where they originated. And RNA interference, also known as RNAi, all started with petunias. In the 1990s, a group of researchers wanted to alter the color of petunias into a more violet color. They thought by overexpressing the violet gene, it would produce a more violet color. Instead, the petunia turned white. It was not discovered until 10 years later that what was occurring was RNA interference. Scientists back then wanted a magical way to silence genes. They did not know at that time that we did not need to practice magic, since every organism, from plants, animals, and humans, have this natural mechanism to silence genes. Over time, scientists realized this and figured that we should take advantage of this natural mechanism. RNA interference is a hot new thing. Studies on RNA interference even claimed Nobel Prizes in 2006 by Angie Fire and Craig Mello. The discovery of gene regulatory RNAs has led to attempts to develop drugs made of RNAs such as silence interfering RNA to silence genes, like the name suggests. These are just many examples of silence interfering RNA drugs from the media in recent years. Because I work in a biochemistry lab, we are interested in small molecules, and enzymes are essential to like a much bigger goal. Keeping that in mind, dicer is the main enzyme of RNA interference, and it is responsible to dice or cleave RNA into smaller RNAs. Products that are approximately 21 to 25 base pairs long, depending on the organism dicer is in. It could dice two types of RNAs, pre-microRNAs, which is also known as hairpin RNAs, and double-stranded RNAs. First, it could cleave pre-microRNAs into microRNAs, and second, it could cleave double-stranded RNAs into silence interfering RNAs. Here is the crystal structure of dicer. Within dicer, there's different parts of the end domains. What I specifically work on is the helicase domain, which is involved with ATP hydrolysis and RNA binding. Because the helicase domain is because the helicase domain in human dicer is pretty big and is difficult to work with, I use smaller versions of the helicase domain in various organisms, such as the bread mold Neurospora crassa and the thermophilic fungus Sporotrichium thermophiles, also known as spoth. We chose these organisms based on how well the helicase domain is conserved with the human dicer. As I have mentioned before, dicer can either cleave pre-microRNAs and into microRNAs, or can cleave double-stranded RNAs into silence interfering RNAs. Because my organisms have two dicers, dicer 1 and dicer 2 in them, my question is then, which dicer prefers which RNA? Will dicer 1 prefer to bind to pre-microRNA or double-stranded RNAs? With this goal in mind, I seeked out to do the following steps: purify the helicase domain constructs and analyze the RNA binding to our proteins. First, I'll talk about the protein purification. So I took my helicase domain constructs and purified them in bacterial cells. In general, I based my protein purification from the following steps. I started out with cells and bacteria. I break open these cells to get our not so clean protein. The protein is washed through a column. Then lastly, the protein is separated by size, by this sizing column, like the name suggests. in the Dissecting column, bigger molecules will go down faster than the smaller ones, since the smaller ones get stuck in the pores of the column. Over time, the sizing column will loop samples into a test tube, and as seen above, again, larger molecules will go down first. Halfway through, our purified protein will be eluted since it's not too big and it's not too small. And at the end, smaller fractions will come out of the sizing column. From this special column, a computer program produces a chromatogram for each protein purification, giving us a readout of what gets produced at a certain elution time. So here are the results for dicer-1 helicasamine for both organisms. This is a typical chromatogram of a protein purification. The x-axis measures the elution time, and let's say the y-axis measures the amount of protein for simplicity's sake. And I would like to note that the bigger peaks correspond to more protein being generated. From the chromatograms, we saw that both, when comparing, let's see, sorry. Okay, so, is this it? Yes. From the chromatograms, we saw that when comparing dicer-1 in both organisms, there is a drastic change between both organisms, the N-casa and the S-dermophile. As you can see from the start, that little to no protein was generated for N-casa. In contrast, the S-dermophile, we generate relatively high yields of protein. We see similar results for dicer-2 in both organisms. Again, Rna casa, it generated little to no protein, while S-dermophile produced relatively high yields of protein. So because of these results from the protein purification and the low yields for the Rna casa, we eventually moved on to choose S-dermophile to continue with our project. Okay, so next I'll analyze the RNA binding to our purified protein using a gel shift assay, which I basically did this entire summer. Okay, so the gel shift assay is to study the interaction between RNA and protein. The first step is to radiolabel the RNA in order to visualize the RNA protein interaction on a gel. Let's say, for example, that there's a double-stranded RNA. One strand would be labeled first before it interacts with the protein. Next step, we'll combine both the protein and the RNA into a reaction tube. Next, these interactions will be run on a gel. If the experiment is successful, one can see a nice titration curve as seen here. From there, we can quantify the bands on the gel and get the KD, also known as the dissociation constant, using a computer program. So this is an accumulation of my long months of doing gel shifts with different truncations of dicer and different lengths of RNA substrates. I quantified the bands on the gel and computed the respective KDs as seen above. I would love to talk about all the gels that I've done this summer, but I'm going to present two gels that I believe generally represents the two stories that I'm going to try to tell. The first story was centered on pre-microRNAs, and my second story was centered on the double-stranded RNAs. Here is a typical gel of my pre-microRNA binding, with the pre-microRNA control lane on the far right, and here is the RNA protein interaction, as seen in the gradient triangle, from lowest concentration to the left to the highest concentration to the right. So I quantified these gels and calculated protein bound to RNA over the total and produced a graph using a computer program. It is important to note that the KD and the affinity has an inverse relationship, so in layman's term, the bigger the number, the less dicer binds to the RNA. And see from the graph, with the higher graph above, spot dicer-1 helicasamine has a better affinity to pre-microRNAs than spot dicer-2. And here is a typical gel of my double-stranded RNA. For dicer-2 helicasamine, we see some binding to the 22 base pairs double-stranded RNA, but not sufficient enough to quantify the gels. In contrast to dicer-2 helicosamine, there is no binding for dicer-1. So those were my typical gels that I encountered this summer. From the summer, I found two general trends of dicer-1 helicosamine and dicer-2. First, we saw dicer-1 helicosamine binds very well to pre-microRNAs, as you can see in the orange and the red graphs. Second of all, we found that, in contrast to dicer-1, we found that dicer-2, which is in blue, prefers to bind to double-stranded RNA than pre-microRNAs. So if you see this graph at the bottom, the dicer-2 helicosamine binds very well to double-stranded RNA. It does bind well to pre-microRNA, but not as well as dicer-1. So in the end, we accomplished our project goal in the beginning and found that which RNAs dicer-1 and dicer-2 bind to. So you may be wondering, like, why are you interested in this and why is this significant? The experiments I've been doing until now have been in a pursuit to understand the way dicer dices. When one understands how dicer cleaves the RNA substrates and which RNA it cleaves, one is able to understand how to silence the genes. And in the long term, one can take advantage of dicer's mechanism of dicing and can theoretically turn off a gene of a disease. So in summary, we found that S. dermophile, also known as SPA, is very well behaved in bacteria, and we definitely will work with this organism in the future and will attempt to crystallize dicer with an ideal RNA substrate. I would like to take this time to thank Jennifer Doudna, who allowed me to work in her lab, Marianne Kidwell, who is my graduate student mentor in the Doudna lab, who guided me through learning how to pipette, to teach me how to radio label RNA. She was truly a great mentor. She even came to lab even though she got into a bicycle accident. And the entire Doudna lab for all the advice and support. I see some people back there. It has been such a great summer. You guys are probably the best lab ever. And I don't want to forget Nathan and Leah for all the help for this conference. And of course the CERF sponsors. I would not be here if it wasn't for them. And